Hello everyone, thank you for listening to this webinar. I'm Mikhail Dozmarov and I'll talk today about how to explore the genome and our experimental data within it. The title of my talk is on your screen and I'll be talking about GenomeRunner, a software that allows us to do just that, genome exploration and exploring our data within the genome. Let me first reflect on 10 years of genome research. As we all know, draft of human genome was created in year 2000, and it was found it has about 3 billion base pairs. Human genome contains from 20 to 25,000 genes, and it's been found that about 1.1 to 1.4 of the genome sequences code for proteins. But the genome is not just a sequence. The genome contains quite a lot of interesting elements, like enhancers, bidirectional promoters, highly conserved regions, and quite the opposite, regions that are organism-specific. And information about such genomic elements we can learn from different sources. The main source of genome annotation is ENCODE project. ENCODE stands for Encyclopedia of DNA Elements, and ENCODE project provides us with information about non-coding sequences, regulatory elements, conserved and low complexity regions, epigenetics marks. To add true complexity to this, ENCODE project provides us with genome organization in different tissues and cell types, in different phenotypes and diseases, and in different organisms as well. This graph on your screen shows how fast we accumulate genomic data. Here, in the year 2000, that's when human genome was fully sequenced. And since then, the amount of genomic data and genomic databases and resources skyrocketed exponentially, and there is no sign of stopping it. This graph shows pretty much the same. It shows the amount of identified single nucleotide polymorphisms within human genome. And it grows exponentially and no way of stopping it. The latest SNP database contains more than 41 million SNPs, identified single nucleotide polymorphisms. So the question is, what are we going to do with this large amount of data? And here is the problem. Genome is big, but the number of genomic features and interesting genomic elements make exploration of the genome and experimental genome-wide data exponentially bigger. And so far, focus has been on analyzing genomic features one at a time. For example, are my set of SNPs affect protein coding genes? But the question remains, what is the functional significance of our experimental genome-wide data in light of all available genomic features? A year ago, BioIT World magazine published a note, The Road to the $1,000 Genome, and they emphasized this problem from another angle. We actually coming to $1,000 genome, and soon we will get it. But the question remains how to interpret this data. And we may end up, we will have $1,000 genome, but it will come with $20,000 analysis price tag. We have great tools for genome analysis. We can visualize genome-wide data along with genomic features and interesting genomic elements. We can detect functional consequences of sequence changes. We can, for example, identify whether a SNP affects protein coding regions in a very bad manner, so the protein won't be functional. We can perform homology comparisons, align sequences. We can perform phylogenetics analysis and study evolution. But these are always for looking at individual and local genomic features. The question I'm addressing: can we analyze our data globally and across all available genomic features? And the answer is yes, now we can. And I'll tell you more about Genome Runner software, which has been designed to make sense out of our genome-wide data. But first, let me remind you how shall we orient ourselves within the genome. We all know the genome is located on chromosomes, and each genomic feature has coordinates. We have to know where, in which chromosome genomic feature is located, where it starts and where it ends. And by genomic features, I mean any genomic elements, genes and exon boundaries, single nucleotide polymorphisms, transcription factor binding sites, various methylation marks, DNA's hypersensitive regions, CpG islands, you name it. Let's make more formal definition of genomic features. Genomic feature is a genomic interval with defined properties. Example, transcription factor binding sites. We know where transcription factor binds, and we should know in which chromosome this transcription factor binding site is located, where it starts and where it ends. We can visualize those different genomic features on one screen, and UCSC Genome Browser is probably the best well-known genome browser for visualizing different genomic features on one screen. And let's have a look at the example of UCSC Genome Browser. Different genomic features are very well organized on one screen. Even this screen looks a bit busy. It's quite logical. Here you can see highlighted a track for a gene, and on this track you can see how a gene is organized, where it starts, where it ends, how many exons it contains, and where exons are located, where do they start and where do they end. And we see many other tracks. For example, transcription factor binding site track. We can see where different transcription factor are bound. But the problem remains. Visualization is local. We can visualize only part of a human genome at one time. And you have to know which genomic features to consider. There are many genomic features, and this screen shows only just a part of it. But actually, UCSC database contain more than 6,000 genomic features, genomic feature annotation tables. ENCODE data occupies the most part of it. Genes and exons, they are also coming from different sources. UCSC has its own gene annotation, RIF-seq has its own gene annotation. There are other gene annotation sources, transcription factor binding sites. There are lots of data about different transcription factor binding sites, both experimentally identified and computationally predicted. Lots and lots of data. And all this data is available for download. All 6,000 tables can be downloaded and used. And Genome Runner uses those tables. I will tell you later how. We learned about genomic features, genomic interval with defined properties. And we learned that genomic features are located on chromosomes, they have start, they have end. Let's define feature of interest. And feature of interest is experimentally generated genomic intervals of interest. For example, ChIP-seq data. We perform ChIP-seq experiment. We run our data through some programs and identify regions of interests. And those regions of interest, they are of course also located on chromosomes, and they also have start, they also have end. So this how we can correlate our genomic features of interest with features of interest experimentally derived in one space. And here come experimental questions. How our features of interest correspond to genomic features? And first of all, we can perform simple association analysis to answer a question. Are my experimental features of interest overlap with genes, with promoters, with transcription factor binding sites, or maybe with other genomic features? Just are they overlap or not? And another question, if your features of interest overlap with genomic features, are those identified overlaps statistically significant as compared to those by random chance? Genome Runner was designed to answer both of those questions, and you can see schematic diagram of how Genome Runner works. You have to provide Genome Runner with input, and your input is a set of features of interest. You have to provide coordinates of your features of interest, at which chromosomes they are located, where they start, where they end. When you give Genome Runner your set of features, Genome Runner looks for UCSC data tables and imports them into local database, just to speed up the process. And Genome Runner performs annotation and enrichment analysis, just as I mentioned before, and from annotation analysis, we get a set of annotated and prioritized features of interest for downstream validation. From enrichment analysis, we get p-values of enriched associations. And the goal of both of those analyses is to understand the biological meaning of our genome-wide data. Let's have a look at annotation analysis more closely. So annotation analysis is designed to answer a question: how do your data related to the whole genome? The idea behind annotation analysis is very simple. First, it's to better functional annotation of GWAS data. If we have our features of interest, we would like to know which genomic features correlated, co-localized with your feature of interest, overall, globally. And based on this knowledge, we can perform genome-wide prioritization of features of interest for downstream analysis. The input for annotation analysis is one or a set of features of interest. And the output, you can get a summary and detailed descriptive statistics about all genomic features corresponding to a feature of interest. And you can get a summary score reflecting the number of features of interest associations with genomic features. Co-localization tests, how do they look like? Let me get the pointer and show here. This purple highlights our features of interest, and blue highlights our genomic feature annotation track. And we can have many situations. We may have situation when our feature of interest lay outside of genomic feature, so there is no co-localization. We may have a situation when our feature of interest co-localized with genomic feature exactly at the middle or somewhere in between. We may have a situation when our features of interest just touches the end of genomic feature, but this is the situation when feature of interest is counted to be co-localized with genomic features. Now, what about regions? So far on the previous slide, I showed you features of interest. They are very short, one base pairs, like SNPs, for example. What about regions? Regions, how to define regions? Regions have length. They are longer than one base pairs. Genomic features are regions. Genes and exons and upstream promoter regions, transcription factor binding sites, they are all long, they have length. Genome-wide data, beside single nucleotide polymorphisms, besides SNPs, can also be regions. For example, ChIP protein DNA binding sites, they also located on the genome and they have length. If you study microarray probes, probes are on the microarray, they are also long, 50 base pairs, 75 base pairs, but they have length. And analysis of regions is much more complicated. Just to give you an overview how many situations we can encounter with analysis of regions, this is very busy tables and you don't have to look very closely at all of those situations, just to give you an idea what you have to deal with. But Genome Runner can deal with all those situations with equal efficiency, and annotation analysis can provide us with summary and detailed information about genomic features overlapping with experimental features of interest, and from this knowledge we are able to prioritize experimental regions of interest for further investigation. Let's say you have 100 regions of interest and you find that one of them correlate with transcription factor binding site, with DNase hypersensitive site, with several methylation marks. So this region, just by the number of co-localized genomic features, is quite interesting to consider for further investigation. Now, the more interesting analysis, enrichment analysis. And enrichment analysis can tell us an answer. Are those overlaps, those co-localizations of our features of interest, statistically significant? The idea behind enrichment analysis is to find statistically significant associations of GWAS data with all available genomic features. And enrichment analysis allow us to answer questions like, is my set of features as a whole, co-localized with genomic annotation features more often than could happen by random chance? In input, you provide a set of features of interest. It's not about analysis of a single feature of interest, it's about a set. What's so special about your set as a whole? And as an output, you will get p-values of enriched associations. For example, you have a set of 100 SNPs or 100 ChIP regions. And Genome Runner found that 60 of them fall within alternative splicing sites. The question, is it significant? And to answer this question, we perform null hypothesis testing. A null hypothesis is tested that a set of SNPs and the same number of randomly selected points within a genome are associated with genomic feature with the same frequency. To test this, we perform Monte Carlo simulation or random sampling, in other words, and proper background, in other words, a universe from which we perform random sample, is very essential. By default, our universe is the whole genome, but this situation may change, and this is described in Genome Runner's tutorial. Let's have a look at proof of principle, what Genome Runner can tell us. About a year ago, a paper was published about critical associations of non-coding RNAs with introns. First of all, non-coding RNAs, those are RNAs that do not code for proteins. They do not code for physical representation of information into them. But they are present and they are important. And those authors identified associations of non-coding RNAs with introns. On your screen you can see a picture of typical gene. And typical gene has exons. Those regions contain information that are encoded into proteins, and the gene contains introns that are spliced out when mRNA is translated. And the authors are identified that non-coding RNAs are associated with introns. So I asked the question, can Genome Runner identify the same associations? To answer this, I looked for statistically significant enrichments of more than 5,000 non-coding RNAs from Affymetrix Human Gene 1.0 ST array. And what Genome Runner told us, first of all, let me show you the full screen of this slide. First of all, Genome Runner identified that non-coding RNAs are associated with genes. So they are associated with genes. Where in the gene they are? Genome Runner tells us that non-coding RNAs are underrepresented within exons. So if they are within genes, and if they are not within exons, so they should be within introns, pretty much as the authors identified. Genome Runner provides us with new information. It identifies that non-coding RNAs are actually also associated with DNA's hypersensitive sites, statistically significant. So after seeing all those red and green lines and all this over and under representation, you may wonder, is Genome Runner identify something that is not significant? The answer is surely yes. And if we test association of non-coding RNAs with transcription factor binding sites, we will not find any statistically significant associations. So, Genome Runner identified known associations and found new. Let's have a look at another situations. What enrichment analysis, what's new enrichment analysis can tell us? And if you go to genome.gov website, you will find catalog of GWAS studies, and you can find different SNPs associated with different diseases. So from this site, I took 141 type 2 diabetes SNPs and 61 systemic lupus erythematosus associated SNPs. Both of those diseases are autoimmune related, but of course there are different diseases. And the question is, what's so special about SNPs associated with one disease and SNPs associated with another disease? For 141 type 2 diabetes SNPs, Genome Runner identified statistically significant enrichment within genes, exons, transcription factor binding sites, and H3K27 trimethylation mark. And this H3K27 trimethylation mark is a mark of repressed genes. So from this, we can hypothesize that in type 2 diabetes, canonical promoters of repressed genes are affected. What about lupus? 69 systemic lupus erythematosus associated SNPs. Genome Runner found them to be enriched within 3 methylation marks. H3K4 trimethylation is a marker of active promoters. So that can tell us about genes that are actively transcribed. H3K4 monomethylation is a marker of transcription enhancers. And the third mark, H3K27 acetylation, distinguishes active enhancers from inactive. So from this, we can hypothesize that in lupus, actively transcribed genes are affected, and this happens via epigenetics mechanisms. We can go downstream and identify in which genes those SNPs are located, in which distance they are located, and what are we going to do with this information down the road. So that's the way to go with it. Now let me tell you a little bit about exploratory analysis, where no man has gone before. We can do quite a lot with enrichment analysis. For example, we can find enrichment among genomic features themselves. For example, we have transcription factor binding sites, and we know that transcription factor binding sites are most often co-localized with promoters of the genes. But what other genomic features associated with transcription factor binding sites? What methylation marks? How do they distribute it? What about other genomic elements? Or maybe transcription factors themselves tend to be co-localized. For example, an FKB transcription factor binding site may be co-localized with another transcription factor binding site. Genome Runner can identify this. We can characterize distinct properties of different classes of non-coding RNAs. For example, we all know non-coding RNAs do not code for physical information, but they are there, and there are different classes of non-coding RNAs. What genomic features they are sharing in common? We can take one family of non-coding RNA, test it for associations of genomic feature. Take another family of non-coding RNA, test it for associations, see what's statistically significant, and make conclusions from it. We can identify features associated with virus insertion sites. We all know that viruses can insert their DNA into the genome, and those sites, we can learn what's so special about them. Are they vulnerable in some way, so the virus can target those sites specifically and learn how to prevent it? We can correlate epigenetics marks with alternative and mRNA splicing. We can also find common and unique rules of genome organizations across multiple organisms. For example, we find that in human genome, one genomic feature correlates with another. The obvious question to test is this correlation hold in mouse, in Drosophila, in C. elegans, in any organisms. And this we also can learn about genome evolution, how everything was developed. To conclude my talk, genome feature can be explored for statistically significant associations with your user-provided genome-wide data. Association and enrichment analysis of experimentally provided genome-wide data can provide understanding about underlying biological processes. And exploratory analysis allow us to reveal new understanding of genome biology across multiple organisms. Now, Genome Runner is an open source software, and it can be obtained free of charge from those web addresses. Application about Genome Runner has been submitted to Bioinformatics journal, and it's very easy to operate. Right now, Genome Runner designed for Windows platform, and it runs as a GUI, and it's very straightforward. First of all, you give a name of your job and select your background. Default it's the whole genome. Then you provide Genome Runner with files containing coordinates of your features of interest. It's simple tab delimited plain text files. Then you select which genomic features you want to analyze your features of interest against. For example, you may correlate your features of interest with genes, with alternative splicing events, with DNA's hypersensitive sites, or with any of those. You can check the settings, which are described in the very detail in the tutorial, which is also available on previous websites, and you virtually click two buttons. If it's annotation analysis, you click annotation analysis button. If it's enrichment analysis, you get enrichment analysis button. And the results you can also get as separate files, which you can view in any text editor. On this, I have to acknowledge my collaborators, Dr. Jonathan Wren, who in year 2005 outlined basic principles of genome exploration and principles of Genome Runner in general, Corey Giles, Lucas Cara, funding that made this project possible, and please ask any questions you may have. Thank you. I guess you have to type the questions into the chat box to get them to me. Let me just add that Genome Runner project is an ongoing project. It's currently still under development. Right now it works for, if you go for the website, you can download it for human genome, but in about a week or a couple of weeks, a version will be available that will work with mouse genome as well. And the question I got, can we check CGH data? The answer is yes, we can. You have to know where your CGH regions are located, and yes, Genome Runner can provide information what's so special about those regions. Another thing to consider, of course, which genome assembly you use for your coordinates. For human, the latest genome assembly is HG18 or HG19 already. So Genome Runner currently works with HG18. So if you want to convert your coordinates on UCSC Genome Browser web page, there is a tool to do this. But as I mentioned, in a week or two, there will be a new version of Genome Runner that will be able to work with the latest HG19 genome assembly and coordinates in it, and with mouse, the latest assembly of mouse genome. Okay, I guess not very many questions. Anyway, the web address for Genome Runner project is on your screen. You are welcome to check it out, and contact information is available. You can write an email, and I'll be happy to answer all your questions at any time. All right, thank you for your attention.